Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Benefits of Utilizing Early Precision QT and Artificial Intelligence-Powered Data Quality Assessments in Early Phase Trials. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the richest clinical evidence in the industry. Across decentralized, hybrid, and site-based clinical trials, Clario's deep scientific expertise, global scale, and the broadest endpoint technology platform allow their partners to transform lives. Now I'd like to introduce our speakers for today's event. Dr. Vik Patel is board certified in cardiology with a PhD in biophysics and has done extensive research on mechanisms of cardiac arrhythmias. He was on faculty at UPenn School of Medicine for 11 years, where he directed molecular arrhythmia research and practiced clinical electrophysiology. Dr. Alain Gay is board certified in cardiology after completing his training as a cardiologist at the Hôpital Cardiologique in Lille-France. Dr. Gay practiced for 10 years in a community hospital, completing his training in cardiac electrophysiology and cardiac pacing. And Jean-Philippe Couder is a scientist with experience in the development of groundbreaking technologies in the field of quantitative electrocardiography and cardiac safety. He is also an internationally recognized authority in the development of computerized technologies related to cardiology. But without further ado, I'd like to hand the mic over to our first speaker, Dr. Alain Gay. You may begin when you're ready.
Thank you, Ryan. Thank you for having me here. And let's talk with the beginning of clinical research, which is first in human trials. So on the next slide, we will understand that in most cases, first in human trials are conducted in healthy volunteers based on the non-clinical evidence which has been provided before by their colleagues. It's an essential step. It's the very critical step, and as beginnings are always delicate times, we have to acknowledge that we have to give great scrutiny and great quality to these trials, because these are the first time that human beings are exposed to a given new molecular entity. And we also have to appreciate that in some cases, unfortunately, more so than we would like, they are the last point where these new entities are being brought to clinical attention, because they have to be stopped for an unmitigatable safety signal or poor pharmacokinetics. Now, on the next slide, we will understand what are these kind of studies. So they are defined or often use single ascending dose or multiple ascending dose study design. What does this mean? This means that we expose a small number of participants to get information on safety and tolerability, pharmacokinetics, and try to identify, whenever possible, the maximum tolerated dose of the entity that we are looking at. The difference between a SAD and a MAD study is that in a single ascending dose study, you only use one dose of the drug per group, where in the multiple ascending dose study, you give multiple doses to the same group of participants. You usually, of course, begin with the single ascending dose before moving into the multiple ascending dose. On the next slide, you will see an example from the literature of how that can look like from a study design point of view. So you see that in the blue boxes, you have dose level 1, dose level 2, so on and so on. And at some point in time, a dose will be chosen by the sponsor and their advisor to move into the multiple ascending study, where you can, like it says, study different doses in the same group of patients. You usually recruit 6 to 8 participants per cohort, usually having 2 on the placebo, which means this is a 6x2 randomization scheme. We will look at the further details on the placebo just after, but for now, that gives you a sense of the magnitude of patient that we would like to, or in that case, healthy volunteer that we would like to engage with. And you see also that you can run these studies in conjunction or in parallel to other studies, like, for example, sometimes you have differences in sex, or you want to investigate the food effect if your drug is given pairs, or you want to investigate different formulations or different drug-drug interaction, which is a major component of drug exposure. So like I said, in the next slide, we will ask ourselves the question, should there be a placebo group? So the usual answer is yes. Why is that? The placebo group will give us a more robust data set that will help the conversation with the regulatory agencies in due course. And there was a small example where it's really helpful to have a placebo group, because in that example, we had 2 subjects who had actually drug-related cardiac events in that double-blind placebo-controlled first-in-human study. Sorry. However, we understood then when we unblinded the data that these both subjects were actually in the placebo group. So the new drug was not to be involved at that point. In the next slide, please. How can we make it a little bit more efficient? Because you would say, okay, I ran my SAD study, then I ran my MAD study. Well, you can actually merge them into an overarching or umbrella protocol, which gives you the opportunity to have only one protocol for the 2 components of these SAD and MAD studies. So this approach is, of course, time and cost saving, can give you additional information because you can put all your data together as one bigger database, which you can use for further analysis if that's needed. And that gives you also an opportunity to adopt adaptive principles for decision making when you want to look at each individual dose and know what to do, how to progress or to stop a dose regarding the data which are collected during the course of that study. In the next slide, we will understand that what is really crucial in this first-in-human studies are the cardiac safety aspects. Unfortunately, the vast majority of the adverse signals that we see in this first-in-human are cardiac-related. And we want to avoid having false positive signals. Therefore, we need to have the greatest quality, meaning having sort of the purest evaluations, if you like, so we can make the right assessment and provide to the regulators and the sponsors with the best safety profile that can be achieved at this point in time. So of course, we need to ensure that the data is collected in a correct and a reliable way, especially in the higher dose cohorts where this is where you are likely to see the truest signals, because of course in the lower dose cohorts, you want to start with dosages which are supposedly harmless, if you want, so we can draw better conclusions. So in my last slide, let's wrap up and understand that first-in-human studies or study is a critical point in time to determine drug success. These SAD and MAD studies are the first in humans. They can, and they often more than the opposite combined. They are designed to investigate the maximum tolerated dose and explore safety for the healthy volunteers who are exposed, and especially cardiac safety. And it's very important to have high quality data and a full complete data set, so informed decisions can be made by the sponsors for the benefits of the patients in the end of the day. I would like to thank you, and I will hand over to Dr. Patel, who will help us with the ICH E14 guidance and the early precision QT. Thank you.
Thank you, Dr. Gay. In this next section, I want to go over how the ICH E14 guidance developed, and along with that, how early precision QT also evolved and how it can be useful to get high-powered data to help us determine the cardiac risk in early phase human volunteer trials. Now, the E14 guidance evolved from the fact that it was known for quite some time that QTc prolongation can be a surrogate of lethal ventricular arrhythmias such as Torsades de Pointes. This was known since the 1960s, but it wasn't until Seldon came on the market as the first non-sedating antihistamine for treating allergic rhinitis that a small excessive but real incidence of sudden cardiac death was noticed. Now, obviously, patients with allergic rhinitis are normally fairly healthy, and so having them die suddenly was obviously unacceptable with the benefit-risk ratio. So this led to withdrawing Seldon from the market in 1997, but it also led to the development of the E14 guidance, which now required all drugs to undergo thorough QT testing, and then this was published in 2005. This led to cardiac safety and QT prolongation being the main reason why drugs were abandoned in development or withdrawn from the market. And then about 10 years later, the IQ-CRS study led by iCardiac, which is now a part of Clario, showed that thorough QT-like results could be obtained from existing first-in-human trials with the standard single ascending, multi-ascending dose designs. And then this was also updated in the guidance December 2015. Go to the next slide, please. So the E14 guidance has evolved through several regulatory updates and question and answers. As we said, in 2005, the first guidance was released, but then in 2009, the first question and answers came out to address several questions, those being methodologies for how the ECGs be collected and analyzed, the difference of gender QT in males and females, the use of a positive control in study designs. In 2012, the second question and answer came up, which further addressed questions about ECG methodology and QT and gender, but also addressed how to assess QT in late phase trials. The third question and answer came out in 2014, where now the requirement for doing a thorough QT study for large biological molecules, specifically monoclonal antibodies, was removed, as these molecules don't have off-target cardiac effects. But also guidance was given around study designs for patient populations where healthy volunteers could not be used in certain indications, specifically oncologic trials, where you could now just have patients without the need for positive and negative controls. In 2015, the fourth question and answer and revision 3 came out, which was largely updated to include concentration QT effect modeling as a way to assess the risk of QT's prolongation for mild effects. And then in 2022, there was a combination document which released the preclinical cardiovascular safety guidance, the SB7 document, with the clinical QT guidance E14. And this guidance gave outlines for the best practices for conducting the preclinical studies, the in vitro hERG assay and in vivo QT studies, and that when these studies were done to high standards, they could be used to do an integrative approach with the clinical data so that the exposures didn't have to be super therapeutic. if there was good quality and margins on these preclinical studies. We can go to the next slide, please. So we now know about 10 years later, after revision 3 in 2015 to the E14 guidance, that phase 1 ECG data can be used in place of a dedicated thorough QT study. The majority of drugs that cause QT prolongation per Sodder points do so through a concentration-dependent effect on the QT interval. And so the exposure response analysis that we call concentration QT modeling is a biologically appropriate way to assess this relationship. Also, getting assessment of the QT prolongation earlier in the development in phase 1 has several advantages. The first being that the sponsors can acquire definitive data earlier on in development, rather than having to wait to do a typical thorough QT study later in development. So this can inform portfolio decision-making decisions earlier. The data can be acquired within the standard phase 1 SAD/MAD study design. You don't have to do any modifications to the study, collect the data. The additional cost of collecting ECGs and analyzing them is just going to be a fraction of doing a dedicated thorough QT study later in development. And now these approach of using concentration QT modeling to assess QT prolongation and real prolongation is well accepted by all regulatory bodies. Go on to the next slide. Now I'm going to take the next few slides to go over the IQ-CRS study and what it told us. So the objective of this study was to evaluate whether assessments of the QT interval in early phase clinical trials can be used to either augment or replace that coming from a traditional thorough QT study. So this was a prospective trial conducted in healthy volunteers that took advantage of the standard phase 1 study design. It was designed in collaboration between the IQ Consortium, the Cardiac Safety Research Consortium, and the US Food and Drug Administration. There were 20 male healthy and female healthy volunteers enrolled in the study, and they were treated in 3 treatment periods. There were 5 study drugs that were chosen had a positive effect on the QT interval. And I'm sorry, the slide doesn't seem to be showing them, but they were ondansetron, quinidine, dallansterone, moxifloxacin, and dofetilide were the 5 drugs that have known to prolong QT interval. One drug that was known to not prolong the QT interval, levocentrisine, was chosen. And then there was also a placebo treatment group. The trial conducted with 2 dosing days. On the first day, the target was to get exposures to the point where they could increase the QT interval by 10 to 12 milliseconds. And on the second dosing day, exposures were tried to achieve to increase QT intervals by 15 to 20 milliseconds. The ECGs were analyzed using the EPQ method developed by iCardiac at this point. We can go to the next slide. So the results of the trial, when they came out, were reviewed and independently confirmed and analyzed by the FDA. They were then presented to the E14 discussion group and endorsed for inclusion into the revision 3 update in December of 2015. So from that point on, the union intersection test, which was the standard by time point analysis that was the analysis used for the thorough QT study, along with concentration response analysis, was accepted as either method for ruling out or assessing QT prolongation in drug development. The criteria that was set, that was the upper bound of the 90% confidence interval could not exceed 10 milliseconds with either analysis. And more importantly, the requirement for a positive control was also excluded with the concentration QTC analysis. We go to the next slide. So this shows some of the results that came from the IQ-CRS study. Moxifloxacin had the largest effect on QTC interval, and what's shown here is on the x-axis is the exposure plotted in nanograms per milliliter. On the y-axis, the change from a placebo-adjusted change from baseline in the corrected QTC interval. And you can see this shows a very positively increasing slope for moxifloxacin. The error bars of most of the time points exceed the 10 millisecond margin, which is shown as the dotted second line on the graph. And so this is a drug that has a positive effect on QTC interval prolongation. You can see here in the bar below that on study day one, the exposure was achieved about 1,800 nanograms per ml, and the predicted increase in QTc interval was almost 15 milliseconds. If we go to the next slide, we show the results for levocentrizine, which is a drug that doesn't prolong the QT interval. And again, concentration of levocentrizine in nanograms per ml is on the x-axis, placebo-adjusted change from baseline and the corrected QT intervals on the y-axis. And you can see the response curve here is very flat. None of the confidence intervals exceed the 10 millisecond upper bound, as shown by the dotted line above there. And the flat response shows a negative effect on QTc interval. Again, the exposure on day 1 was about 1,000 nanograms per ml, but the predicted increase in QTc interval was only 2 milliseconds. We go to the next slide, please, Ron. Now, I want to talk a little bit about the QT methodology and how we can actually improve precision. And I want to point out this will have to be used mostly in healthy volunteer patients, where the QTc intervals and the ECG waveforms are well defined. For patient intervals, we have to still use the traditional triplicate extraction method. But in healthy volunteer studies, both methods will record continuous 12 ECGs. The data is then recorded either on an SD card or uploaded directly via the internet to the ECG core lab. And then with the traditional method, an analyst will visibly and manually view the data in the extraction window to find the best stability of the RR intervals, where you can extract 10-second ECGs. 3 10-second ECGs are usually extracted from the extraction window, and then 3 beats on each ECG are measured for the RR and the QT intervals to give you 9 QT-RR pairs. With the EPQT method, the data is also collected with continuous 12-lead ECG recordings, again uploaded SD card or to the internet, where you can then go to the ECG core lab. But now the iCompass automated algorithm will automatically identify the stable RR intervals within the extraction window. And then 10 ECGs are extracted from the extraction window that are 14 seconds long. And then an automated combination approach using iCompass with human oversight will identify all usable RR-QT pairs that can be analyzed. And this usually leads to 100 to 120 QT-RR pairs that can be analyzed as opposed to 9. We can go to the next slide. So what this does and why this makes EPQT so valuable for healthy volunteer studies is that you have much higher number of data points to give you higher precision to determine a smaller effect on the QTc interval. Again, it was designed, as we mentioned, for using continuous 12 ECG monitors to extract 10 ECG replicates at each time point. It's an automated approach using human oversight so that all usable ECG RR and QT intervals can be used. The approach provides higher precision data at each time point because you get more time points that can be analyzed from even a smaller study design with fewer patients in it. That's typical of a phase 1 study. So the key advantage is that the higher precision that's provided by EPQT allows the study to provide higher power to detect a smaller QTc effect with the same sample size. And then this reduces the risk to the sponsor without the additional cost or risk of modifying the study design. What we often do with this is that we'll store the data in an early phase trial. We know this trial will progress. So we call this a collect, clean, and store approach. And often the data will just sit there and you don't know if the data is usable or it has too much artifact on it. And so to help alleviate that risk, we've developed a method where we can actually look at the data before it's analyzed. And that's what Dr. Kuderek will discuss next on the next slide, our AI-powered tool to assess data quality.
Thank you, Dr. Patel. Very happy to be part of this seminar. Hello, everyone. And I'm going to talk about this tool we've developed around the utilization of machine learning. Let's go to the first slide. And what I want to say first is that obviously we all are exposed to machine learning every day now. This is a technology that has been really taking over a lot of applications. And in the field of medicine, obviously, imaging diagnosis has been one of the applications that has been pioneering really the application of machine learning into medicine. And it has really demonstrated an ability to do things that humans are not able to do, either in terms of identifying patterns or generating useful information based on large databases. But the application of machine learning is not limited to imaging. And what you're going to see in this presentation is that we actually decided to use machine learning and this powerful tool to do signal processing. The signal processing, ECG signal processing, is in general a set of tools that are linear processing blocks that are put together to extract pattern and information from signal. By using machine learning and deep learning, we now push these analyses in nonlinear equivalent type of tools that help us really handle a much broader set of problems. And again, the one that we're going to tackle here with this technology is the rapid assessment of the quality of ECGs that are actually submitted to Clario. And this is basically a tool that we've built over a certain number of months, but really we put together in Clario, and I'll show that in the next slide, a set of experts. Can we go next slide, please? Thank you. A set of experts that really combine different type of discipline. We have, obviously, in addition to Dr. Patel and myself, people that have experience in putting technology into, I would say, drug safety assessment, and we have a set of unique experts in development of software, management of software, and development of signal processing tools. So I wanted to acknowledge our team, and really we form at Clario a team for advanced computational methods to cardiac precision science. And this group of individuals, as you're going to see, have developed a very, very unique tool for our organization. Let's go to the next slide. So I'm going to talk about convolutional neural network, which is usually presented and is a multi-layer neural networks. But let's formalize that a little bit so it is hopefully clearer to everyone. The so-called CNN is part of different type of families of machine learning. So if you put some kind of hierarchy around the formalism of this technology, there is a whole category of tools that are called artificial intelligence, that include machine learning, that include deep learning, and the convolutional neural network are part of this deep learning type of technology, where you have an input layer where you put information in this input. You have multiple layers that I'll show in the next slide are usually combining all this information and learning about what the model should do. And you have output layers that gives you an output, a quantitative output for obviously a certain purpose. And here it's going to be around quality. Let's go to the next slide. On the next slide, I wanted to say that the method to develop this kind of machine learning is pretty standard, but very interestingly, the FDA recently released a good practice, I would call it, for developing machine learning. And we have followed actually this recommendation when we did this work. There are several aspects that are very, very important. First, there are actually 3 steps of development of machine learning. There is a training step where basically you need a large amount of data to have the machine learning to learn about what you want to do. You have what we call then a tuning step, where basically you expose new data to this model and you tune in this model to do even better than in the training. And you have a final step, which is called the validation step. And what is important in this final process is really to apply the model to independent data and new data to really validate the approach and make sure that what you actually came up with in terms of performance in the 2 first steps are actually confirmed in the 3rd step. What is very unique to machine learning, and most of you may have heard about it, is that the more data you expose to this system, the more the system learns about the functionalities they have to do. It is a very important actually aspect of machine learning, because it means that, again, the more data you use, the better your system is supposed to actually developed in a new system. So the FDA really developed a way of how to record these changes, how to make sure that when you change actually your model over time, you do it in a way that it indeed lead to better performance rather than over learning or having a performance that goes down. So the FDA recommended different things. This is obviously more for medical device. The technology I'm going to present does not require clearance from the FDA, and yet we decided to really follow those good laboratory practice. Let's go to the next slide. So there's a little bit of a difference between the general machine learning and deep learning. Again, what we've developed use deep learning. The difference is actually around feature extraction. What I mean by that is that when you do machine learning, and it has been published in many papers for many type of applications, specifically in ECG, is that you may use from the ECG interval measurement, QRS duration, QRS amplitude, QT interval, PR interval. So all these features of the ECG that you can first measure and then feed your actually method to learn or to do whatever you want to do with this signal. In deep learning, the approach is a little bit different, where actually you don't have to create feature extraction. You can actually feed the model with the basic raw data, which is the ECG signal. And that's exactly what we've done in this application, where instead of putting any layer of measurement on the ECG, we get rid of this aspect and we feed the machine learning algorithm with ECG, with the pure ECG. One of the constraints when you do these kind of things is that you have actually a constant number of inputs, meaning that when you're going to feed this algorithm, you need to make sure that the duration of the signal that you feed the model with is always the same, which might create a problem because we know that heart rate changes in human. Hence, for a certain duration, you may have different number of beats, you may have some changes. So what we did, and I'll show that on the next slide, we actually developed the concept of ECG epochs, which is actually a piece of ECG that is 3.6 second length that is centered on one single bit. This centered bit is going to be really where the information are going to be analyzed, because when we expose this 3.6 second to the model and we have millions of those epochs sent to the models, the heart rate will make the bits around the central bit moving around, and this information will be blurred. So in a sense, we actually use this interval of 3.6. I'm going to refer to it as epochs. And we basically use this information to learn what we wanted to make the system learn what we wanted the system to do, which is actually to recognize whether this ECG is a good ECG or is a poor quality ECG. How do we define that? So we define that based on the 3 bits on lead 2 methodology, the 3BL2 methodology, as you may have heard about, that actually use measurement on 3 consecutive bits on lead 2 in the ECG strips. At Clario, we have gathered millions of these epochs over the years, and we are using this data set of ECGs to really feed this machine learning. And obviously, all this data have been manually reviewed and has been labeled good or bad, or poor, I should say. So we have a unique set, a very rich database of all this ECG, and we really built on this database to develop this convolutional network. What we define as a poor epoch is one actually set of bits where we were unable to measure the QT interval due to inadequate quality. Okay, so we have this million type of epochs. We have this data fully annotated. So we have now the data to make the machine learning and the CNN learn about the quality. How do we measure this quality? Ultimately, as you can see on this schema, we input the ECG signal, and we output one single score that varies from 0 to 1. 1 means that the quality is high and is good. 0 means that the quality is poor. Okay, let's go to the next slide. So the ability to computer to process a lot of data is just amazing today. So once we develop this model, not only we can apply it to an epoch, but we can apply it very quickly to a large number of epochs. We can apply it not only to 10 hours continuous signal, but to a 24-hour, 72-hour ECG. And the way it works really is that we are running now continuous ECG, extracting every possible epochs from a continuous ECG, and have our machine learning model generating a quality score for each of these epochs. Again, as I said, the higher the score, the better the quality. And you have here an example that shows you how basically we decompose this continuous ECG for one single lead and generate for each of these epochs a value. Let's go to the next slide. So how do we apply that in the EPQT context? Here, this is actually a figure that shows you all this calculation done by the machine learning algorithm. It's a lot of data. So what we decided to do is really to use a color scheme to really report this information. What you have on this plot is 3 levels of representation of this data. At the top, you have what is a 10-minute time point extracted from a study. So the beginning of the figure on the left is the beginning of the time point, and on the right is the end of the time point. What you can see on this upper panel with all these colors is that the green represent actually a value of the scoring, quality scoring close to 1, meaning good quality. When the color goes into the reddish or orange values, it means that the score is close to 0. So what you have here on this upper panel is a representation of the quality of your time point. And here it's 10 minutes of continuous ECG. And you have the ability to really visually find what are actually the part of the 10 minutes where the quality is the best. In this example, we also put in blue where in this specific study, the 10 strips were extracted from the time point, okay? And this is an example, for example, this is the example of strip number 9. So on the center panel, you have the machine learning quality score that is reported again using this color scale for the selected ECG strip. And as you can see, this is not all fully green. There is some bits that are associated with a little bit darker color, meaning that those bits are not as stable as the other. Visually, it's not obvious, right? You can see that the T-wave, for example, here is a little bit changed in terms of morphology that generate these changes in stability of the signal. So very interestingly now with this machine learning algorithm, we have the ability to very quickly review time points and a very large amount of data in the data that are submitted to Clario. Let's go to the next slide. This is another example that again emphasizes how we use the score to identify bad quality data. You have at the top our color scheme for reporting the quality score, and on the lower panel you have the tracing, the ECG tracing, and you see where we have noise and abnormal bits. We have actually a darker color that again identify the area from a time point, from an extraction strip, where the ECG is low quality. Let's go to the next slide. So let's go into development of this model. So far I assume that we had the model and everything was working, but let me show you how we developed it. And it was quite a large amount of data that we looked at. We obviously follow, as I said in introduction, the training, validation, and test set. We built this model on more than 350,000 epochs ECG. Those were collected from 118 studies. So 118 studies were included into the overall development of this technology. We used 24% of the data for training, 3% for validation, and 74% for testing. Importantly, I'll say that the studies were common between training and validation, meaning that some of the studies that were presenting in the training or used in the training were also used in the validation. Some of the participants, because this learning is at the epochs base, it's not at the subject base or study subject base. So really, some of these participants were also common between the training and the validation. But when we did the test, we used completely independent studies with completely independent participants. And here, as you can see, we validated the model on almost 260,000 epochs ECG. So this is really a very unique data set that we had to develop this technology, and you'll see in the next slide that the results are very impressive. So we use this technology for a couple of things, and the first one I wanna present is around labeling of ECG epochs, right? So we taught the model how to assess the quality of these ECG epochs, okay? In the validation, and the numbers that you see here are the results for the validation, the independent data set. And as you see, we were able to reach 93% of true negative and 97% of true positive. What are true positive and true negative? True positive are the bits that are actually containing a good bits and that are detected as good by the AI model. The true negative are these epochs that are poor quality and were actually detected as poor quality by the model, okay? So really, the model classified 215,000 of the epochs as good, 97%, and 34,000 of the epochs as poor, 93%. And those were absolutely correct. Again, those poor and good quality ECG were based on the 3-bit on lead-2 methodology. Let's go to the next slide. We actually extended then the application of this technology to detecting 3 valid ECG replicates in a given time point. Now that we can, with this technology, know which bits are valid for doing measurements, we can now combine those core and say, okay, what are actually the number of time points that have at least 3 valid ECG replicates? And we tested that in 3 studies, 2 SAT studies and 1 MAT study. As you see here in this table, you can see the number of subjects per study. And more interestingly, you can see our ability to really being able to say whether those replicates are available for each time point. The true negative here were 100% for all the study, right? Meaning that when 3 valid ECG replicates were not available, we were actually able to find them. The true positive here are in general above or close to 98%. Again, here, the technology tells you, oh yeah, this time point, you can do your extraction of replicates, no problem. And in general, 98% of the time we were correct using this technology. So this is very interesting application, obviously, because now we have a tool that very quickly, and I would say almost in real time when we receive the data, can process all these millions of bits and tell us what's happening in the data that were submitted to Clario, and really have a report that tells you the data that you capture and collected during your study is good enough to do actually what you want to do with this data. Let's go to the next slide. So this is how we integrated the artificial intelligence technology into the EPQT process at this point. It is part of the collect, clean, and store service. Again, repeating what I just said, now with this layer of artificial intelligence tool, every piece of data that is submitted by a sponsor to Clario to this collect, clean, and store services is automatically analyzed, and a report is generated to provide information about the quality of this data and the ability of extraction of a replicate ECG for each single time point of the study. This can be extended to more than that. Obviously, since we do the measurement of all the ECG, we can have scoring system that tells you really what is the quality of the data that has been captured in your study. We believe this is a very, very valuable set of information that now is, again, available in this service of collect, clean, and store, and almost real time, I would say, in a sense. Let's go to the next slide. So I want to conclude now. Can we jump to the next slide, please? Thank you. So I want to conclude now and really emphasize that we develop machine learning, a machine learning tool to assess the quality of continuous ECG tracing collected in early phase clinical trial. And we have a tool that is very close to real time today. We already perform a manual review of a small subset of data in the collect, clean, and store study to provide assurance that the collected data will be usable for future analysis if required. This is actually a very important aspect of the value created by this technology. We improve data processing speed and data accuracy as well to assist in the interpretation of data, promising exciting future application in this domain. At this point, as I described, this is actually fully integrated into the collect, clean, and store services. The next step with this technology is to push basically this assessment of quality more and more into the EPQT process for two objectives. The first one is to reduce the need for data review, human review, and the second one is to at least attain the same quality of accuracy and performance of the technology, and hopefully even doing better by applying this new algorithm that tells you where the good quality data should be extracted from your large set of information. So we are excited about this technology, about this application, but even more excited about what's going to happen in the future and how we're going to leverage this amazing technology to make Clario services and help sponsors to really have more confidence in the data they use in their clinical studies and ultimately the data they submit to FDA. I would like to thank you for your attention, and I'll be happy to take some questions.
Well, thank you very much for this very insightful presentation. As we move on to the Q&A, I'd like to direct the audience's attention towards the handouts module within the GoToWebinar control panel, where they can download some additional material related to today's presentation. I'd also like to encourage the audience to keep sending their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, I've already received some questions, though, from the audience, so let's get started with those. And Vic, I've got a question here for you. Someone's wondering, are the proposed solutions able to analyze the transmural heterogeneity of QT?
Well, thanks. That's a very good question. And the direct answer is that it cannot. Transmural heterogeneity is really looking at a difference in repolarization between the epicardium, mid-myocardial layer, and the endocardial layer. We're just using surface 12-lead ECGs for the solutions. So we don't really see the heterogeneity between the layers, but that does get reflected in the surface ECG by the QT prolongation when there's heterogeneity between the layers. So we're looking at the longest leads. So the answer, it's not direct, but it is indirectly assessed when we're using all 12 ECG leads.
All right, wonderful. Thank you so much for that answer. Next question here, and I think Jean-Pierre, perhaps you can share some insight on this one. The question is, did all epochs come from ECGs generated from the same ECG machine?
In a given study, yes, obviously. So in a sense, there's enough standardization, I would say, around ECG recorders to feel confident that our model applies to different type of recorders. I have no doubt if we are talking about bedside ECG. I'm not talking about ECG patches or this kind of devices. But for the one that are used in phase 1 clinic and in general in clinic setup, there's a pretty good standardization of what these machines are supposed to collect and how they collect it. So I have no doubt that the model wouldn't be impacted by ECG that would be generated by recorders coming from Philips, GE, or the Baxter type of ECG device companies.
Right. All right. Very good. Now, so how about for poor ECG epoch? Do you distinguish abnormal ECGs from poor ECG images?
Repeat this question. I'm not sure I get this question.
Sure. Someone's just wondering simply, it says for poor ECG epoch,
Yeah.
do you distinguish abnormal ECGs from poor ECG images?
So first, we don't use ECG images, right? We use ECG signal, okay? Abnormal ECGs, we have to define what it is, right? I mean, when we say poor ECG, you can imagine that if you have ventricular ectopic beats, arrhythmias, first, obviously, this shouldn't happen in the extraction, but if it was there, obviously, it would be really affecting the quality score, right? So in a sense, do we discriminate between them? I would say no. I would say no. I would say that if we had this kind of arrhythmia, it would actually impact the quality score. Yes. If it's the question, I'm not sure I responded well to this question.
Yeah, well, we can, thank you very much for that answer. And of course, we can follow up with that person after our webinar. As another question here, and perhaps Elaine, you can answer this question for us. The question is, so SAD and MAD studies discussed today were in healthy volunteers. Someone's wondering, what about oncology?
Thank you, Ryan. That's a very good question. So indeed, in oncology, it's because of the unmet need for the patients, it's usual not to do this kind of study in healthy volunteer. And the second reason is the toxicity of the drugs that are investigated in oncology. So you don't want to expose healthy volunteer to this kind of toxic drugs. So therefore, the studies are mostly conducted in patients. So that's the main difference. And a little bit to what Jean-Philippe said about the quality of the tracings, this also affects a little bit the assessment in a way that unfortunately, these patients who have cancer sometimes are also older people who have comorbidities, including heart disease that may affect the signal which is collected. So that makes them a little bit more complex, but just a little bit more challenging as well.
Perfect. Thank you so much for that answer. Now, Vic, I wonder, can you explain for someone the main difference, the 5.1 pathway and the 6.1 pathway in the latest ICH E14 update?
Yeah, thank you, Ryan. Again, that's a very good question from the audience. So the differences are a little bit subtle. Both of them require that you have negative results from the in vitro hERG assay and the in vivo QT study that don't show any risk for prolonging the QT interval. But with the 5.1 pathway, the data can have smaller margins. That is, you don't have to have as large a margin of 50-fold for the hERG assay, and you don't have to reach the high clinical exposure in the preclinical model. Again, animals have different responses to exposure than people do. So you may have to actually get to exposures that are 2 or 3 times higher than they are in patients. And so if you don't have that in your preclinical data, you can get the 5.1 pathway approval by just getting the clinical exposure in your clinical trial. And that will be a label that says that you don't have a risk of a large QT prolongation. If you do meet the 50-fold margin with the hERG assay, and you can get to the high clinical equivalent exposure in the preclinical QT in vivo study, and then you get to clinical exposure in your clinical study, then by the 6.1 pathway, you can actually get a label that says you don't have a risk of proarrhythmias on the label. So it's really a labeling issue and how kind of the margins you have in your preclinical data is the difference.
That's great. Thank you so much for that insight. Now, Jean-Pierre, I have a question for you here. Someone's wondering if you did you compare your AI model with other tools for ECG quality assessment?
Well, Yes, in a sense, to what we had currently as an ECG assessment. We have another way in our processes to measure the quality of ECG at the bit level, which is based on signal-to-noise ratio and a more standard approach around the quality of ECGs. And we did actually look briefly, I would say, around the correlation between our machine learning scoring and those more linear assessment of quality. And we saw some relationship, but we didn't really push this investigation further, I would say. But there was some obvious association between those approaches.
Perfect. Thank you so much. Vic, another question for you here. In the future, do you imagine your model to replace ECG machines to assess cardiac function and detect abnormalities?
Yeah,
Well,
thank you, Ryan. I mean, that's a good question, right? And this is the question everyone has around AI models and what they can do. For right now, the model is really just looking at data quality. And I think we will not be using this model specifically for doing ECG analysis, but there is development both at Clario and other places where that might be happening in the future. We have to understand that has to be well validated, right? So if you have a model that can analyze an ECG and function, which is already being done by some other companies, the question is, though, it has to be validated and well accepted before it can just be replacing interpretation by a physician or a medical personnel. So I think the answer to the question is, our model will not do that, but development in that space is happening and will continue to happen, both at Clario and other institutions.
Excellent. Thank you so much for that answer and for all of the answers today. However, we have reached the end of the Q&A portion for our webinar. For those interested in hearing more from Clario, though, you can register for their next webinar, Going Beyond ATS/ERS 2019 Guidelines for Your Respiratory Clinical Trials. And this is scheduled for Wednesday, April 18th, and you can use the link that I'm sending in the chat box right now. Also, if we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you.
Thank you.
Thank you.